Yes. Okay.
I hear you loud and clear. You don't hear me? Am I muted in too many windows here? I was muted on Zoom, not on YouTube. So let me know when I should get started.
Introduce you to the guys here with us in the morning.
Okay, great. Then let's just get on with the show. So hi, welcome. I'm glad to be joining you here. Even though it's remote, would have been more fun to be there in person. What I'll be talking about over the next half an hour, 45 minutes, is network-based omics integration, analysis, and visualization. And first of all, even though I have been introduced a little bit, let me just get started by saying, who am I? I'm Lars Juhl Jensen. I'm pretty active online, as you've probably guessed, considering that I'm doing things on YouTube. You can also find me on Twitter, so you're welcome to contact me there if you have any questions. And of course, my daytime job is I'm a group leader. I'm a group leader at the Novo Nordisk Foundation Center for Protein Research. And there I run a research group doing computational biology, network analysis, network biology, call it what you want. I'm also a co-founder of a company called Omics, and they're doing the same kinds of things. I'm not working in the company, but I'm obviously one of the owners, and for funding reasons, I'm required to tell you that. It's not to advertise the company. My group is called Cellular Network Biology, and that captures pretty well the vast range of things we are doing, which includes things like working closely together with the mass spectrometry groups next door on analyzing their omics data and trying to make sense of it, so omics data interpretation, and also a lot of work on resource development. Of course, by far the best known of the resources that I'm involved in developing is the string database. So the string database, you probably know it in the form of a website where you can go in, query for a protein of interest, and get a network back of that protein together with other proteins that it's likely to work together with. And that's of course very useful if you have a protein of unknown function and you're trying to figure out what it does. You can also query with a list of genes and get a network back for those, and that way figure out which of your genes are likely to work together, that way sort of grouping together proteins of related function. And that's of course what's useful when you're trying to interpret omics data. The starting point for creating string is a collection of more than 5,000 genomes, which encode almost 25 million proteins. And the goal of string is to link these together with what we call functional associations. And by that we mean we don't just link proteins to other proteins that they are likely to bind to. We link proteins to other proteins that they're likely to work together with, so being, for example, in the same pathway. The way we do it is through data integration. So we're trying to integrate a lot of different types of data, and that includes things like genomic context, and that's particularly useful if you're looking at bacterial genomes where you have things like operons. What are you doing? Let me get over here. Where you have things like operons, and you can then genomic context where you have things like operons and you can link proteins to other proteins based on how their genes are in the genomes. So what we're seeing here is you have the green, the blue, and the yellow gene, and they're sitting next to each other in the top row in what looks like a potential operon. Now, the trick is, of course, just having genes sitting next to each other in one genome may not be much of a signal. But if you go look at evolutionary conservation and look across a lot of genomes, as signified by the other lines here, you can see whether this operon looks like it's evolutionary conserved. And if that's the case, then very likely these proteins work together. If you're interested in eukaryotes, you will more want to look in the direction of interaction data, so physical, so experimental data like co-expression, for example. And you don't have operons in eukaryotes, but you have things like co-expression data, microarrays, RNA-seq data nowadays, where you can link genes to other genes based on which ones are expressed under the same conditions. So the idea here is you're looking across thousands, if not tens of thousands of different conditions, and if two genes show similar expression across such a large and diverse number of conditions, then very likely they're doing something together, considering that they're clearly needed at the same time. We also integrate physical interaction data coming from assays like, for example, tandem affinity purification followed by mass spectrometry, where what you do is you take a protein, you tag it, you do a pull down with that protein, and then you use mass spectrometry to identify what's in the pull down. Last but not least, we're of course happy to take the low-hanging fruit, what we call curated knowledge, and that's basically, it's different from experimental data in the sense that this doesn't mean that somebody's done one experiment. It means textbook knowledge. This is the established things that we know about molecular biology, things like physical protein complexes that are well established, or pathway databases where we have all these diagrams of whether it is signal transduction pathways or here metabolic pathways, where you know which proteins are involved in the same processes inside the cell. And of course, it's a ton of work making these databases, but thankfully other people are doing that. So we say thank you, we download it, and we import them into String. So that, in a nutshell, is the String database. But as you probably imagine from the length of the presentation, it's a little bit harder than that. First of all, there are many databases. I said we have pathway databases. We have a lot of pathway databases. We have a lot of different databases of physical protein interactions. We have many different databases of genomes. And we need to somehow pull that data down and put it together. These databases have an ugly tendency to come in different file formats, and even if people have standardized the file formats, we still have the problem of different identifiers. So in the same file format, different databases will still use different database identifiers for the same genes and proteins. The data are of what I very politely refer to as varying quality, which means that some of the data is god-awful, and the data are just fundamentally not comparable. How are you supposed to compare predicted operands to physical interaction screens to manually curated pathways? It's just fundamentally not the same thing. So some of this is just hard work. There's no two ways of putting it. When you have a lot of different databases, somebody has to download them. When they come in different file formats, somebody's going to have to write a lot of parsers. When the databases use different identifiers, somebody's going to have to make mapping files. All of this is an awful lot of work, but there's not too much to say about it because it is really a lot of manual work. It's what I like to call the bioinformatics equivalent of pipetting. Where things get a bit interesting is when it comes to dealing with the quality issues of the data, where the first step is to develop quality scores. So that means that for any given type of data, we try to come up with a scoring scheme where, given that kind of evidence, we can take protein pairs and rank them from which ones are most likely to be correct to which ones are least likely to be correct. If we go back to the tandem affinity purification data as an example, what you have here is you have 4 different pull downs that have been done. We tagged the blue protein, we saw the green and a couple of others. We tagged the white protein, we saw the blue and the green in the pull down together with it. We tagged another white protein, we saw the blue but not the green. And we tagged the green protein, we got a pull down that didn't contain the blue. And now we want to somehow calculate a number that allows us to say, how sure are we that the blue and the green protein are in a complex together? And while it's not clear how exactly one should do that, I hope it's obvious to you that the 2 pull downs on the left are positive evidence, and the 2 pull downs on the right are negative evidence. So, of course, the more often we see the 2 proteins together, the more likely we are to believe that this is a genuine interaction, and the more often we see pull downs with one but not the other, the less likely we are to believe it. If you think about it in a bit more detail, imagine we've done a big pull-down screen. We've done a lot of pull-downs. We know how many we've done. We know how many contain the blue. We know how many contain the green. We know how many contain both. That's a 2x2 contingency table. Based on that, you can calculate a lot of different scores. You could do a Fisher's exact test to get a p-value. That was the first thing I tried. That turned out to not be a very good score. You could calculate an observed over expected ratio. That's a better idea. It works better. You can do even better than that, but that's not really the point. The point is you have to understand the data, you have to be creative, and you have to come up with a scoring scheme. And you have to do that separately for each kind of data. Because obviously the way you score, say, co-expression data is going to be completely different from how you score physical interaction data. The next thing we do is then take these different so-called raw quality scores and calibrate them on a common gold standard. And what we use as a gold standard is pathways. We happen to use the KEGG database, but that's not really important. You could have used a different database. And now whenever you're looking at two proteins, if we just look at, for a moment, the proteins that we actually know what are doing, so the ones that are assigned to pathways, whenever you're looking at a pair of proteins, there are two options. They're either in the same pathway or they're in different pathways. So now, if I take all the interactions from this pull-down experiment and I bin them based on their raw quality score, I can look and say, let's look at all the pairs scoring between 1 and 1.1. How many of them are true positives in the sense that they are on the same pathway, as opposed to false positives being in different pathways? And it turns out the ones scoring between 1 and 1.1, it's something like 14% of them that are in the same pathway, which tells you that in this case, a score between 1 and 1.1 is a pretty bad score. You can look at the ones scoring between 2 and 2.1, and you'll see it's something like 80% of them that are in the same pathway, which tells you that a score in that range is good. You do this for a lot of different bins, you fit a calibration curve through it like shown in this figure, and now you have a function that allows you to go in and take a protein pair where you don't know the truth. You can then score it based on the data. It gets a raw quality score of, say, 1.7, and if you go in and look up on this plot, you will now find that a score of 1.7 corresponds to a 50-50 chance of the two proteins being in the same pathway. So basically, that evidence boiled down to a probability of 50%. You have calibrated, even though you have different raw quality scores for each kind of data, if you're looking at this and you have these calibration curves, you can now turn everything into what is the probability of these two proteins working together given this one piece of evidence at hand, whatever it is. So what we've accomplished here is to put everything on a common scale. So that's the good news. We've taken all this data, we've done all the data integration, we've scored everything, we put it on a common scale. The bad news is that even after doing all of this, we are still missing most of the data. And you might wonder, where's all that data that I'm missing? Well, that's where we get to text mining, because where the data is hiding is here. This is a back-of-the-envelope estimate of the biomedical literature. If we naively assume that everything is indexed in PubMed, and that behind each of those 30 million entries in PubMed, you have a 5-page article, and you print that out on standard 80-gram A4 paper, you get a pile well over 10 kilometers. Of course, not everything is indexed in PubMed, and the average paper is for sure longer than 5 kilometers, and the pile is for sure taller than 20 kilometers. Now, this doesn't matter. It doesn't matter whether it's 10 or 20 kilometers, because the reality is that in either case, we cannot read it. There's just too much. Even if we didn't have to read the existing literature, we couldn't even keep up with reading the new papers as they come out. Because currently, you have a new paper coming out in PubMed about every 40 seconds, every day of the year, all day around. So, we can't read the literature. That's the harsh fact. And because of that, we have no choice but get a computer to do it for us. And whenever I need to get a computer to do something like that, that even halfway approaches being smart, I find it useful to think about the analogy that a computer is about as smart as a dog. And by that I mean, if I put sufficient effort into it, I can teach it to do specific tricks. So, borrowing a cartoon from Gary Larson, what we say to dogs. Okay, Ginger, I've had it. You stay out of the garbage. Understand, Ginger? Stay out of the garbage or else. And the only thing the dog hears is blah blah Ginger, blah 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 Ginger, blah 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 blah. It understood its own name. And that's all. And this is the task that people in text mining call named entity recognition. Finding the things in the text. In our case, it would be the proteins being mentioned, but not understanding the rest of the text. And unsurprisingly, when you want to do named entity recognition, the key ingredient in doing a good job with that is to have a good dictionary of the names you want to recognize. So you need to know that there is a protein called cyclin-dependent kinase 1. You need to know that CDK1 is the same thing. You need to know that CDC2 is also the same thing. Then you need, on top of the good dictionary with all the names, you need a blacklist or a block list or whatever you want to call it, a list of stop words, a list of the names that are fundamentally a bad idea for doing text mining. So these are not names that are necessarily wrong per se, but they are names that from a text mining perspective are a spectacularly bad idea. My favorite example is that the Human Gene Naming Committee thought it was a good idea to make the recommended gene symbol of a gene this. Now, anyone who's ever been in a wet lab will know that SDS is a commonly used reagent. It's a detergent, it's something you use for denaturing proteins. It happens to also be the recommended gene name of a gene, but of course most of the time in the literature, people are talking about SDS, the detergent. So if you're text mining with a dictionary and you think that every time the text says SDS, they're talking about that gene, you are going to be wrong a lot. And of course, this is particularly bad when the next step is to do relation extraction, where we go in and look at which genes are mentioned together. So this is the simple strategy for pulling out relations from the literature. You simply say if two things are mentioned together often, then they likely do something together because people probably don't mention them together for the fun of it. So what we're doing concretely is score-based counting. It is in some sense pretty similar to what we did for the pull-down data. We look at how often are A and B mentioned together compared to how often they are mentioned separately. And the trick here is we don't just count in a naive way. We actually take into account the distance, so we know that when things are mentioned in the same sentence, it's more informative than if they are mentioned just in the same paragraph, which again is more informative than if they're mentioned in the same paper, but not even the same paragraph. We are also working a lot currently on developing deep learning techniques, where we're using a lot of these modern machine learning techniques to both improve the named entity recognition and to pull out specific relation types like physical interactions. Lastly, we of course do score calibration again, so we take these text mining scores, we calibrate them against the gold standard of KEGG maps, just like we did with all the other data, so that we can put it together with everything else, and that way get it into string. So that's the network part of string, but another side of string that has become increasingly popular is using string for doing enrichment analysis. You probably heard about it already, otherwise I'm sure you'll hear about it later in this course. Enrichment analysis is something you very often do. You've done your Omics study, you've done all your statistics, you ended up with a list of significantly regulated genes. You now take this gene list and you compare it to a genome-wide background and you identify statistically significant terms. By that I mean terms that appear surprisingly often for the genes on your list compared to what you would expect given the frequency of the same terms in your background set. And what might those terms be? Well, what every tool does, including string, is of course things like gene ontology terms, pathways. You could also add in protein domains, things like that. But in string we're going beyond this in a couple of ways that is really part of the reason why string has become popular for doing these things compared to a lot of the competition. And one is enriched publications. So the idea here is that we've done text mining, we've done named entity recognition. So we know for every paper, for every publication, which genes are mentioned in that paper. And that of course means that every publication is effectively a gene set. So you can now do the enrichment and say, which papers mention surprisingly many of my genes? Why is that interesting? Well, you might want to go read those papers. The other thing we've added recently is enriched network modules. So instead of going with the annotations from UniProt of Go terms, pathways, all of that, we're taking the entire string network, the complete network for an organism, And then we're running clustering on it, hierarchical clustering, to get a whole hierarchy of nested modules in the network. And then we do statistics on those and find out which are enriched. So you can go and upload your entire dataset from an Omics study, have it mapped onto the string network, onto the pre-clustered string network, and then you can identify parts of the network where this module within the red circle contains surprisingly many of the genes near the top of the list of your genes, or on your list of genes. The other thing that people love to use string for is to do network visualization. So again, this is something you see a lot in omics studies, in particular in proteomics papers, is that people have done an omics study. Again, they have the gene list that they use for doing enrichment analysis. And now they take this gene list and they put it into string, they get a string network, and now they have their figure for a publication. Because showing a long list of genes is boring. Showing a network of all your genes, well, at least it's a figure. And I'm not showing this particular figure from a paper in 2015 to make fun of the authors. This is an example of a network where you can't see anything from this network, can you? But again, I'm not doing this to make fun. There are loads of these. If you just go 2 years later, somebody published an even bigger network where you can't see anything. Add another 2 years, you get an even bigger network. I mean, what's the point of doing this? I don't know it. This is what people in the network field call ridiculograms. And as the name suggests, it's because we think they are ridiculous. You can't see anything out of a network like that, where it's just a hairball with everything connected to everything. And of course, the bad thing here is not just are these not very useful figures, but every time people publish a figure like that, I kind of feel it's partially my fault, because they're using my tool to do it. So the constructive way of going about this is, of course, to think, how can we help people make better figures? And that's when we started working together with the Cytoscape team. So Cytoscape is a tool, it's a network tool. It allows you to work with networks, visualize networks, all those things. It allows you to do network analysis. It allows you to visualize your data to the networks. What Cytoscape is not is a database. Cytoscape doesn't come with a network for you. Cytoscape is a tool for working with networks, but you have to provide the network. Of course, what we wanted to do was to give people an easy way of getting string into Cytoscape. And turns out that's not hard, because Cytoscape allows you to code apps. So they're like plugins, like you know, for your web browser that you can install into Cytoscape to add functionality. So we started working with one of the core developers, John Scudder Morris at UCSF, on developing the stringApp. And that app simply allows you to easily retrieve string networks and have as much of the functionality as possible from string inside Cytoscape, while having the full power of Cytoscape to visualize your own data onto the networks. Which, sort of at first step, allows you to make figures like this. You might argue, this is still a ridiculogram, but at least it's now a ridiculogram where you have your data mapped onto it and you can see from the color of the nodes what goes up and what goes down. And the power is, of course, you have other apps. You have apps like Cluster Maker 2 that allows you to run clustering on the network and now identify network modules, like we did for the enrichment analysis, and use that for laying out the network in a much nicer way. And now when you start doing things like this, you suddenly begin to be able to see things. You have network modules and you have your data mapped onto the network, allowing you to see what is actually regulated, and this can really be helpful for identifying biological stories. Also, it does allow you to still do the enrichment analysis, and once you retrieve the enrichment analysis into Cytoscape, you can again take that and map it onto the network. So now you have the network, you have the nodes, and these halos, the circles around the nodes, allow you to show which proteins have which terms which were identified as significant in your analysis. So the light blue corresponds to one term, the dark blue corresponds to another term. We've also created a companion app called Omics Visualizer, which is designed to handle even more complex omics data. So the idea here is to be able to handle things like phosphoproteomics data that gives you position-specific information. So you have information about each individual phosphorylation site rather than the protein as a whole. It allows you to work with time course data, And of course, what these two things have in common is they have multiple values per node. And that presents a problem when you want to do data visualization. Because if you have a network of proteins and you're now trying to map onto that the regulation, you have the problem that you have multiple different values for one node. So which color are you going to give the node? Well, what you want to do instead is to do something like this, where you have one or more rings, where in this visualization, each ring corresponds to a different pairwise comparison of two samples where you did up-down regulation, and the different segments in the circles correspond to the individual phosphorylation sites. And since the different proteins don't have the same number of sites, they obviously don't have the same number of segments in the circles either. So with that, I was a bit fast. I'm happy to take questions from people first. And then also after that, I'm happy to dive in. I can demo stuff both in string online on the website. I can demo things in Cytoscape, whatever you're up for. But before I take your questions, I want to thank the people who did all the work here. So if you're looking at the protein networks, a lot of this work, string database, Damian was doing so much of the work over the years. That includes when he was a PhD student in my group. Now he's in Christian von Mehring's group in Zurich. So string is a collaboration between Christian's group, Pier's group, and my own group. We have the text mining side of things where many people have contributed over the years. At the moment, Katerina is doing all the work on deep learning together with Sampo Pysalo in Turku. And we have on the Cytoscape stringApp side of things, we have Nadja and John Scudder Morris that I already mentioned, who've been the main two developers of that. And Mark was the main developer of Omics Visualizer. And last but not least, I want to mention the fact that we have an open postdoc position, application deadline, October 15th. So if you're interested, go for it, apply. I only just got sound now, if anybody said something.
The other sources, like experimentally derived sources, I am aware that they have very different scoring. Do you have an idea how they compare with one another? Which one you can actually be more certain is better source, or how do you usually approach when you generate a PPI? Do you just include everything, or how do you approach this?
So in terms of which ones are most reliable, that's the whole point of the score calibration, right? Even though they have very different scores, we're comparing everything to a gold standard, which means that if you put a certain confidence threshold in string, the interaction should be equally reliable at a given score, irrespective of the kind of data that gave rise to that score. Because the score is effectively the posterior probability of these two proteins working together, given that piece of data, whatever it was. In terms of what to use, I would say in most cases, I use all the evidence, because you need a good reason to throw away some of the evidence. You're definitely going to be worse off by just removing something. The exceptions are, of course, there. There are cases where you don't want to include certain types of data. So that might be if you are working on something that is itself derived from the literature and you want to compare it to string, then it would of course be a circular analysis. If you also have the text mining results of the same literature coming into string, then you're going to see agreement between your data and the text mining data based on you and string looking at the same literature. That wouldn't be a good idea. Sure.
For instance, PPI in humans, I imagine that they have a much lower coverage than you can. But for those nodes that you have there for, for the same as similar as it can be, these two networks, have you compared how string improves?
I haven't compared those specifically, but I think the main thing to be aware of is that these are very different networks. So it's not really a matter of which network is better, it's more a matter of which network you want to use for what. Because the reference network that you're talking about is a physical protein interaction network. So it purely captures which proteins bind to each other. Whereas what string is trying to capture is which proteins work together, which is an altogether different question. So of course, if you're interested in physical interactions, well, a physical interaction network is better. If you're trying to group proteins into which ones work together, a network that takes into account the broader landscape of evidence for functional association is obviously better. So it's more a matter of using the right tool for the right job than a matter of saying which tool is better. Yeah, I'm going to take the questions from Zoom first, since those are the people who are actually supposed to be on the course.
With five different types of cancers, then it is very hard for me to see all the publications related to particular proteins if I study to compare with all five different types of cancers. But I was thinking that if I learn maybe text mining technique so that I can sort out the publication related to the protein synthesis.
I had a very hard time hearing that question. Could someone closer to the microphone repeat it?
So please, to repeat it, please correct me if I'm wrong. His question is whether or how you can use string to find the most relevant publications associated with a given protein of interest or phenotype, is it? Yeah. In the example, it was in cancer.
So I would say finding the most relevant literature for one protein is not really something you would use string for. The whole idea of, I think this relates to the enrichment analysis I was talking about. And the way it works is that you have a set of proteins of interest. So you would be taking a set of proteins and then you would run the enrichment analysis. Let me just share a screen here. Multiple proteins. I could take. Do I take? Let's just see if this one gives something. So if I run this analysis here, and I go to the analysis results, so I just queried with 4 proteins, and if I go down to the part called reference publications, you now get the papers that are most enriched for these 4 genes. So you would see that you have a paper here, for example, that mentioned all 4 proteins out of just 11 genes in the list. So that way you can very quickly dump in a list of genes and find papers that talk about many of them. I don't see a way of how you would use string directly to prioritize for a single gene. You could of course imagine that you're making a lot of assumptions here, that you're interested in papers that talk about that gene together with other genes that it works with. So if I went and queried for, this is probably going to be a terrible example, but let's do it nonetheless. If I query for BRCA1, yes, I want it in human. I get a network of BRCA1 together with its friends, so to speak. And I can look at the analysis results here, and I can see which are the reference publications that mentioned a lot of the proteins, not just BRCA1, but BRCA1 and its friends. But I'm not sure if I would argue that those are the most relevant proteins for, most relevant papers, for that one protein of interest. It really depends on your question.
for the person who does that, what information, what rules for the fast information, any information should they expect the type of what the information data to bring?
So uploading an interaction data set is a bit of a different story, right? Because normally you would upload a gene list. So typically the kinds of experiments people come with is more like a perturbation experiment or a case control study where you're looking at which genes change between two conditions or change over a time course. So they don't have interactions themselves. If you're talking about people having done pull downs, then of course you can also have everything from your baits. So you have a bait, you do a pull down, and then all the preys that were pulled down together with the bait, you dump those into string to show it. And you can absolutely do that. So it allows, the neat thing is that especially if you have some quantitative data from the pull down, so if you did pull down in mass spec, right, then you will know how abundant the protein was in the pull down, preferably even compared to some sort of control. And that way you can go in and then fairly easily do the analysis. Let me see if I have one sitting around here.
Yes, but you what you do physical versus function, what do you think?
I mean, you would want to compare to both, but the point is, of course, if you're comparing to physical interactions and you're doing a physical interaction experiment yourself, then if you only believe the things that are supported by existing pull-down experiments, you're not going to find anything that's new, right? Then you only trust the physical interactions that you found if somebody else found them before. That's kind of a problem. So if your concern is that you have a pull down and you don't necessarily trust everything you see in the pull down, and you're trying to find some things that are at the same time trustworthy and novel, then you could use functional associations to support it, because then you're finding things where, according to your experiment, they physically interact, but we have some evidence for them at least working together, but there might not be anybody who's already shown them to bind. Does that make sense?
We have one question over chat on Zoom.
Let me see there. Yes. Right. So I've looked at hive plots at one point. I have to say I was not super thrilled with them. I mean, they look fancy, but it's really hard to see something from it. But the circus plots, you can do things kind of similar to it. I wouldn't do circus plots in Cytoscape, but you can do things that are similar to it, where you do a circular layout of the nodes. So if I hop over on Cytoscape here, what we could imagine doing is, let me just get myself a network. So this is how it works with the stringApp. Let's just take something timely like coronavirus. Let's take something that is not too crazy big, the top 50 proteins, and let's import a network. So now I'm actually doing a text mining search. I'm querying PubMed for coronavirus. I'm downloading the PubMed IDs of those papers. I'm running an enrichment analysis to figure out which proteins are most mentioned in these abstracts compared to PubMed as a whole. And then I'm retrieving a string network for that. So it's a fairly big analysis, and the result you get is this network. Now the power of Cytoscape is that it allows you to do a lot of layouts, and also with apps it allows you to do clustering. So if I wanted to do a circular layout of these, my first step would be to say, let's say cluster maker, let's run an MCL clustering, let's change the inflation factor to make it a fairly fine-grained clustering, and seemingly nothing happened. But if you look down in the node table, I know that you will be playing more with Cytoscape later in the course. You have a new column called MCL cluster that tells you which proteins were in which cluster. So this is now a new attribute. I can now go and say layout, attribute circle layout, lay them out by MCL cluster. And I now have them on a circle based on which ones cluster together, so the ones that are linked a lot to each other are close to each other on the circle. I could now take this one and do things like, where we got it, bundle edges, and it will now try to, where did it go? I'm not seeing anything happen. That might be a Cytoscape bug. Anyway, what it should have done is edge bundling, where it sort of curves the edges so that things that are following the same path are sort of grouped together, making it look a lot more like a circus plot. So yes, you can definitely make them appear on linear coordinates based on whatever you want. So also if you have a time course, for example, you could sort things by the time course. We also have some questions on YouTube. So we have a question about what if you can work with multi-omics data, genomics and proteomics on the same string network. Absolutely, you can do that. Again, I would tend to do that in Cytoscape. So one of the things you can do that is pretty neat in Cytoscape is that you can import your own data. So once you fetch the network based on a gene list, you can now import your data as a table, and that way have it associated with the network. So having it appear as new attributes down in the table here. And you can import multiple tables. So if you have some genomics data and some proteomics data, you can absolutely import both of them. Then, of course, the challenge is how are you going to visualize it on the network? One of the options would be to use Omics Visualizer to simultaneously show the genomics data and the proteomics data. Another question about what do I think about using, say, node size as a way to visualize information. Node size can be a good way of visualizing information, and again, you can do that in Cytoscape. You can use node color to encode things. I think the one important thing to say here is you don't want to be using too many different things at the same time. If you use the size to mean something and the shape to mean something and the color to mean something, you can very quickly overload the user or the reader with information in the figure, producing a figure that is in practice incomprehensible. Cytoscape will happily allow you to do it, but you want to sort of limit yourself in terms of how much you're trying to show at the same time. But you definitely can do things like that. And I think if I just go back, if you look at the slides here, in this network, in fact, you'll see that not only do we use color to encode the regulation of the proteins in this proteomic study, so coloring things based on which expression cluster it was in, but we also use node size to encode information, not quantitative information, but in this case qualitative information, because these samples relate to a particular kind of cancer. And of course, there are some genes that are already known to be important in that form of cancer, and we're highlighting those by making the nodes bigger. Then
We have one more question here.
yes, go ahead.
Time-wise, let's say that you have differential gene expression from 3 different times. How would you know in for a specific set of proteins?
So that's sort of a simpler version of what you're doing here. So you could simply have one of these circles around it, or you can also make things like a pie chart plot. Let me just see, do I have a web browser here? Too many windows, sorry. Screen. If I go to training, if you look at the training material for Omics Visualizer, you'll actually see how to do it, because in exercise 1, we're working with proteomics data, and in that proteomics data you visualize site-specific information, but the second exercise is actually directly about how to visualize some time course expression data onto a network that is not even from string. So there are exercises that we have online on the website that you can just go and do for this. But the short answer is you would visualize it in a way that is very similar to what you see on the slide, where you have different segments of the node or different segments of a circle around the nodes representing the different time points, and obviously having them sorted in a sensible way. The other option, if you're showing very big networks where trying to actually show the time course data itself on each node becomes overwhelming in terms of the amount of information, is to use the time course data to do clustering, identify a discrete set of clusters of these are the different behaviors that I have in my experiment, and then simply use different colors to color them. So that way, doing effectively the same thing as here, where different colors just represent different expression clusters. It all depends on how the message is, it depends on how long your time course is, it depends on how big your network is, and it depends on how nicely things cluster if you run clustering on it. You have to try different things and see basically what makes the better figure. But there are many ways of visualizing time course data. Then there was a question on YouTube about APIs. So there's a lot of APIs available. If you just go to string and go to the help pages, then you'll see that there's a whole API documentation for the string API. So it's a RESTful API, and that of course means you can access it from Python, from Perl, from R, from whatever you want. It's all there. You can also bulk download the data. That's the other option. So you can either access things via API, or you can just download it wholesale and do the analysis yourself. Then there's a question about PubMed, how we prioritize findings, whether we prioritize certain organisms. We don't prioritize organisms, but what we do is that we are aware about which organism we found genes from. And we have to be aware because different organisms, I mean, yeast, Drosophila, are going to have different gene names from human. And when we're integrating the data, we first gather all the data for the individual organisms, and then afterwards we transfer by orthology. So if you're interested in a human network, then of course we're going to trust the results more that come from an organism that is close to human, because we're going to be more sure about doing the orthology-based transfer. But if you go and say, I want a yeast network, then we're going to prioritize effectively the yeast literature the highest, because that's the direct evidence in the organism you're looking at, and we're going to have less and less confidence in the information the further from yeast the organism is. So the way we deal with it is we first integrate at an organism level, and then we transfer by orthology, and in the transfer, the distance matters. So in a sense, it's not directly in the text mining, but in the end result, you do end up prioritizing the evidence based on how closely related the organism is to your organism of interest. Then we have, if I want analysis, phosphoproteomics. So which plugin to help you do analysis of phosphoproteomics, time series data? Depends a bit on what you mean by analysis. If you want to analyze the data, you obviously should be using proteomic software for analyzing the data. I assume you've already done the raw data analysis and you're down to the point where you know your phosphosites that were significantly regulated in your experiment. At that point, what you want to do is again look at the Omics visualizer tutorial on jensenlab.org, because that's the software designed specifically for handling that kind of issues. Does StringDB explore uploaded lists? So when you're uploading, let me see, how is it? This is something where I'm not completely sure. So when you're uploading a list to string, you have the option of letting string keep the list as a list of stuff that was seen together in an experiment. And then that way you can actually also know that some things might be interesting because they were seen together in other experiments. We're not giving away other people's experiments to each other, far from it. And you can choose whether you want us to keep the list. But yes, we do some things like that where you can upload lists of proteins and we can make use of that to analyze other lists of proteins. It's not something I was involved in developing. You'd have to ask the team in Zurich about that. They know exactly how that works. It's a quite new feature. How can I import a network that I already have in string? I'm not quite sure what the question is. So if you're asking how you can get a net, if you've queried in string already and you have the network in string and now you want to transfer it to Cytoscape, if that's what you mean by import, that wouldn't really be the way to go about it. But what you can do is in Cytoscape with the stringApp, you can run the exact same query. And when you do that, you're going to get the same network. The nodes are not going to get exactly the same colors, but again, the colors in string are just to discriminate the nodes from each other. So they don't mean anything. And generally, when I visualize things in Cytoscape, I'll want to throw away the default coloring and make my own custom coloring based on my data every single time. I'm not sure if that fully answered the question. But yeah, the tutorial for that, I missed half of the question there. Again, we have them. If you go to the training, to the training website on jensenlab.org, the Cytoscape stringApp exercises will show you exactly how to do that. I mean, even if you just do exercise 1, you'll immediately see how you do a protein query, and you'll see that you get things into Cytoscape looking very much the same way they look in string. That's a good question. So the question is, if you've done expression analysis, so you have your own expression data and you know which genes are differentially expressed, and now the question is, how can you use string to identify the regulators? So I assume the idea here is that you might not have seen the regulators in your experiment. What you often see are the downstream genes being regulated. And then the question is, can you find the upstream regulators? There's not really a great way to do that at the moment, I have to say. What you can do is, of course, you can take your expression data, you can get a string network in Cytoscape, and then once you have a network, what you can do is things like this. So if I just take this network, doesn't really matter which network it is, I redo the layout to get something that looks a bit better. If I found these genes to be differentially expressed, let's pretend, I know they came from text mining in this case, but let's pretend it was an expression experiment. I can take string and I can say I want to expand my network with, say, another 10 proteins based on them being linked to this. So now I got a set of extra proteins in that are related to the proteins that I found. So that's one thing you can do. Of course, when you find genes that are related to the ones you came with, there's no guarantee that they're going to be the regulators. So you would have to put some filtering on top of that. So ideally what you would want to do would be somehow take your network, expand it to get related genes, and then out of those identify which ones are potential regulators as opposed to things just working together with your genes but not being regulators. It would be a non-trivial analysis. very specific question. How can I color my nodes based on which tissue they're most associated with? There's not an easy way of doing which one they are most associated with, but if you're interested in a certain tissue, then it is very easy to do. So let me just again clean up this network, organic layout. So if you look down in the node table, you will see that there's a lot of columns called something with tissue. And that's data that when you're pulling a network from human or mouse or rat, it pulls tissue expression data from a resource that's also developed in my lab called tissues.jensenlab.org, where you can go in, look up a gene, and it will show you where things are expressed. So this is just to show you where the data comes from. So you see this coloring of the body, these labels, this confidence score, where are things expressed, also goes into Cytoscape, and that's what we have as these scores in the tissue columns. And I could very easily go now and say, let's style this network and say, let's color this based on one of the tissue columns, and say, let's color this based on whether it's seen in blood. Let's do a continuous mapping. I'm not sharing my screen because I'm an idiot. Yes, here's the screen. So let's just go back here. If I undo. So here's my network. You see that there are tissue columns down here. The website I was talking about was this one. So you see I queried for a gene and I got the body. I have those scores down in the node table here, and I can now go and say, let's color things based on whether it's expressed in the blood. I can make a continuous mapping, and now based on this color gradient, I've colored all the proteins based on how confident I am that this protein can be found in blood. Another thing you can do if you want is that you can actually use Omics Visualizer to throw this data on. If you don't want to pick one tissue, but you want to show several, you could go and use Omics Visualizer and say apps, Omics Visualizer, import from node table, take the tissues. Let's just say, I'm sharing screen again. Yes. So here I just went apps, Omics Visualizer, import from node table. I select tissue. I say, let's show whether it's found in blood, whether it's found in liver, and whether it's found in lung. Next, import. And now I could go and say, let's color these. Doing a continuous mapping, let's not put labels on it, let's just draw it real fast. And now you see, I've done this kind of mapping where I've taken each circle and I've cut it up into 3 segments where each segment corresponds to a different tissue. So now I've taken 3 selected tissues and shown all of them in one go on the network. And this is exactly the same tool that I would use for the question asked earlier of how to visualize time course data. This is how you can cut the circles up. You just need to first get the data into your node table in that case, or use Omics Visualizer directly to import it from your file.
Any other questions?
Let me know if I overlooked something somewhere. I think I got through the chat log on YouTube as well.
No? We have one? actually goes through because you could talk, you could like the interaction between the genes different ways. But then you also mentioned that you can take some score based on the this interaction, this show more. My question is, this how to this product. After you put what you into this not a show you like this, very beautiful method. But then what that I can take to really sort it out validations to see so how many of them are really done at different level. I don't think how something confused about how to sequence score etc. So I think we use part of that.
So in terms of knowing which interactions are true or not, there are really only two versions. One is the one that you won't like, which is that you just have to trust the score and say, well, the ones that have the highest score are most trustworthy. And that's kind of what you have to do if you're working with big networks. But if you're working with smaller networks, what you really need to do is go look at the evidence. So if I have a string network and I want to know whether a certain interaction is trustworthy, what you do is you click on the interaction, it shows you all the different types of evidence that are available. And if you want to go look at, for example, the text mining evidence, you can click show and it will show you the actual papers in which it was found. I keep forgetting it. Yes, so if you back, so I just queried for trip A just for the heck of it and get a network from E. coli. If I click on an interaction, it shows me all the types of evidence available for that interaction. And I can then dive in and say, if I don't trust text mining, then I can always go in and say, show me the actual text mining evidence. Show me the text, show me where these names were found together. And you can read the text as a human being and judge whether you think there is indeed a connection. But that's the only way of doing it. For text mining, that's actually the easiest thing to validate because you as a human being, you can read. So you can see if it makes sense. But for the other types of evidence, you're going to be hard pressed to do better than string did, because what are you going to do? You have some interaction experiment, for example. Let's see, do we have something from yeast? Yes. So if I go look at something where I have experimental evidence for an interaction, then it's going to show you which assays it's been shown by. In this case, it's genetic interference assays. And if you go in and then look at the interaction and say, where did it come from? It's going to point you to the paper. And then again, you would have to go look at the paper and see if you trust the experiments that these people have done. Again, you can do that, and we absolutely encourage people to do that. But unless you're really an expert on the types of experiments that people have done, I think you're going to have a hard time doing something that actually works better than the systematic benchmarks that we've been doing in string, where we've looked at which things really work.
No more questions? All right, so I think we are a bit on time.
Yes, that's excellent. Thanks for all the great questions.
Thank you so much. This was really, really nice to hear. I have some exploring to do myself on string. One thing I can say is that even when we generate our own networks, it always ends up with going to Cytoscape. And if we have one more source that we can use to benchmark whatever we found, this is always well. So this will be quite interesting.
So I think one of the last things I might add here is exactly if you have your own network in string, in Cytoscape, and you want to compare it to string, that has actually recently become a lot easier because there's a feature in Cytoscape, and this time I'll remember to share the screen, where I can take a network that I've imported, and then in string there's a feature called stringify. It's grayed out for this network because this network is already a string network. But if you have a network that you've imported and your network is not a string network, then you choose stringify and it effectively takes all the nodes in your network, query the string for those, and creates a string network where it can now get all the edges from string for your set of nodes while keeping your interactions. So you can that way compare your interactions with interactions from string in a rather neat way.
All right, so we thank you once again.
Thank you. Great. Thank you. Bye-bye.